So, we were talking about the various genes that were discovered first in C. elegans, and I wanted to point out again the importance of caspase, SED9. It's a protease. I just wanted to point out that it's a pretty non-specific protease. It chews up just about any proteins. It's dependent on there being a cysteine nearby, and there usually is, and then it actually cuts at the protein where there might be an aspartate. And so that's the name, cysteine-dependent aspartate-directed protease caspase. And it was one of, and so that had been understood, and yet it was still something of a surprise when people found that this was the same, that there were such genes in mammals. So the experiments that were important for this were one in which they grew sympathetic cells in vitro and knew that they had to provide them NGF or they wouldn't survive. And if they removed the NGF from the medium, then the cells would die, these sympathetic cells. But here was the paradox, is if at the time they removed the NGF from the medium, they also replaced it with drugs that block protein synthesis, either to block, just sort of generically block all transcription or all translation. There are different classes of drugs that do that. When they did that, even though there was no NGF around, the sympathetic cells stayed alive. And so that's when they reasoned, it must be that when the NGF goes away, the cell normally starts producing some proteins that are going to cause it to kill itself. And by the way, you might be wondering, what are these drugs that are used to block transcription? They're antibiotics. That's how antibiotics work. Their job is to stop protein synthesis one way or the other, either through transcription or translation blockage. And they do so in a pretty non-specific way, and some of them are very effective. They basically stop all protein synthesis. And so the point of the antibiotics, when you take them to get rid of a microbe, is you're trying to slow down the microbes enough to let your immune system get on top of them. And, eventually you stop taking the antibiotics and protein synthesis, which hasn't been halted entirely in your body, but has been slowed down, then it can go back and you can recover. Anyway, so this told them that cell death, even in mammals, is an active process. That is, the cell has to decide, make some decision somehow, I'm not needed anymore, and then it triggers a set of genes that will then allow that to happen. And so for example, and it turns out that there was not just one caspase in mammals, but at least 9. And they're released in something of a cascade where the first one identified, which I believe was caspase-3, let's see, it was the third one that was identified, hence they called it caspase-3, but it happens to be the one that typically gets activated first, and it triggers the release of other caspases until you finally get caspase-9, which actually does the work. And so for evidence that this is true, it turns out that if you compare wild-type mice at birth, this is what a normal mouse looks like just before birth, with one where you've knocked out caspase-9, and that means that no cells that should have died, and they have these totally overdeveloped brains that won't even fit in the skull, and so they die at birth because there's no covering over their brain. And so you can see here how they've squished as much brain as they can in there because a bunch of cells that should have died stayed alive. And that too, of course, tells you that it's not like those cells were doomed, that they were ineffective or bad at being cells. They can live as long as you stop caspase-9 from killing them. So that was, those were important discoveries. And so here I want you to compare the pathways that are in C. elegans versus mice. So there's this gene EGL1, which was identified in worms, and its job normally is to inhibit SED9, and SED9's job is to inhibit one called SED3, cell death mutant discovered 4, and that in turn inhibits SED3, and it's SED3 in worms that is the real death knell. Once the cell starts expressing SED3, it is doomed, because that caspase is very active, very non-selective, and everything starts getting taken apart. And so that will lead to death. Here we put side by side the homologous genes in mammals. And as is typical when we go from worms to mammals, one of the things that happens is instead of having one gene, there's a whole slew of them in mammals that have evolved, yes, through duplication and divergence. And these genes are, all involved in this process. But the homologies between these particular genes is very obvious and strong. So think about that for a moment. This business of having some cells kill themselves, commit suicide, that's been going on in the development of animals since we shared an ancestor with worms, right? So people had asked in class, why does this happen when you make a cell and then you kill it, even though you went to all the trouble to make it? And again, I don't know why, but clearly it works, and it's been working for a very long time. So EGL1 in worms, there's a equivalent in mammals called BAX, and BAX's job is to inhibit BCL2, which is a homolog of the worm SED9. And BCL2 is anti-apoptotic, right? And so BAX is pro-apoptotic because it inhibits BCL2. And BCL2 in turn inhibits a homolog of the worm SED4 called APAF1 in this case. And APAF1, if it's not inhibited by BCL2, will trigger a cascade of caspases that will eventually lead to caspase-3, which is the one that's closest in function to SED9 in worms. Caspase-3, and again in mammals, once the cell starts expressing caspase-3 to any degree, it's doomed, because it's going to start taking apart the cellular machinery that keeps the cell alive. There's another theme, is another difference between worms and mammals, is in the course of evolution, a whole series of other genes have arisen that are going to play with this process. So one way to compare the difference in the apoptosis between worms and mice is there are a lot more brakes and accelerators in the mammalian line than there are in the C. elegans line. Another indication of how stripped down and bare bones a worm is when you only have 1,000 cells, as opposed to more complex organisms like a person, where there are, billions of neurons and trillions of connections and things like that. Anyway, so among these additions that mammals have, there are a set of proteins that are known as inhibitors of apoptosis proteins. I'm sorry, it's as clunky as can be, but that's what they're called, or IAPs. And their job, they normally inhibit any caspase-9 that's around to prevent it from triggering the cascade that would lead to the executioner, caspase-3. So there's a series of these. And so here's an important break, but there's a break on the break. There's another protein, sometimes called Diablo, which inhibits the inhibitors of apoptosis proteins. So Diablo, in inhibiting the anti-apoptotic proteins, serves as a pro-apoptotic factor. And so you can see, again, brakes on brakes and accelerators, etc. Things get more complicated. And you could probably understand this as there are more factors that can come into the play in the decision about whether a cell will undergo apoptosis in mammals than there are in worms. So more regulation, more ways of regulation, more variety in the regulation, and therefore more variety in the decisions that can lead to apoptosis. And here's a pretty common pathway for apoptosis, in mammals at least. And that is often the final decision to commit apoptosis will result in a release of a lot of intracellular calcium, especially in mitochondria. And so when intracellular levels of mitochondria get high enough, a protein, Diablo, is released from the mitochondria into the cytoplasm. And as I pointed out to you, Diablo inhibits these inhibitors of apoptosis proteins. There's several of them. And so suddenly it is no longer putting the brakes on the cascade of caspases. And if that happens long enough, and eventually caspase 9 gets triggered and done, then the cell is doomed and it will die, because caspase 3 is the executioner in this process. And the BCL2s that we talked about earlier, the ones that are inhibiting apoptosis, they work by inhibiting the release of Diablo. So I do want you to know about that process. Now we're focusing on the caspases, but another thing that happens once a cell makes the decision to die is it also starts making enzymes that chew up the DNA, so that the cell no longer has any DNA, and of course you know that's the death knell. And that fact that the cells start chewing up their DNA has led to one method of labeling dying cells, and that is the so-called TUNEL method, where you put a analog, a synthetic substitution for uridine, and then label the process. So in a normal cell, the enzyme is put here. You put in the enzyme that's labeled with the nucleotide, and in a normal cell where there are no breaks in the DNA, nothing happens. But in a cell that's about to undergo apoptosis, because the cell has chewed up its DNA, now there are all these remaining little tails of various bits of DNA, and there's loads of places for the enzyme that you put over the tissue to insert these markers that you can color, and then you can put it through a chemical reaction like immunohistochemistry or something like that, so that you can see where the brown is are all cells that had chewed up their DNA and therefore are going to die. And so that's one way to do it. Another way that can be done, caspase 3, because it is so specific to execution, there are antibodies that recognize caspase 3 that has been uninhibited, so activated caspase 3, and that's another way where you can get a preview. Oh, these cells are going to die. So the picture that arises in terms of NGF and neurotrophic factors is one like this, where there are certain target cells that produce certain neurotrophic factors. Different target cells produce different neurotrophic factors. And cells that are, that because of their developmental history are dependent on that trophic factor, they take it up, it eventually gives a signal to the nucleus, and it turns off any inclination to apoptosis. However, there's competition between the cells, and so if one cell doesn't get enough of the neurotrophic factor, it will then make the decision to kill itself, dismantle itself, and you're left with a winner. And different targets are competing for different neurotrophic factors, and therefore the competition can be different between them. And sometimes the time at which this competition takes place is different, because remember, it's the neurotrophic dependence of cells. First of all, they're not neurotrophic dependent when they first start, because they grow neurons and send their axons out. So it's a good thing they're not neurotrophic dependent there, because there's no source of neurotrophin, right? But they send their axons out, and then at some point in their developmental history, they become neurotrophic dependent, and they undergo that competition. And so what happens is, as the cells die, those that didn't get enough, you have this pretty even matching of the number of neurons innervating to the number of targets that are out there, which is to say the number of targets that we're producing, producing the neurotrophic factor. So to the question of why go through the trouble of apoptosis, other folks some time ago tried to come up with a list of what some of the reasons why it might be useful to have apoptosis. Maybe somewhat wasteful, but still useful. So to remove cells differently in males and females, and we're going to see that later, where it turns out a lot of sex differences in the brains of animals between males and females are due to the fact that in one sex more cells die in the period of apoptosis than in the others, and you can probably guess hormones often play a role in deciding how many cells will live or die. Another possibility is to get rid of some progeny that aren't needed, maybe for some reason in terms of providing differentiation information to one cell. There has to be one more division. It could be that there are times when some of the neurons are made, but they're not really appropriate. There was a mistake, and so they're gotten rid of. And there, this surely that's a possibility, but there's not much evidence that it's true. There's not much evidence that there are those sorts of mistakes, or that the period of apoptosis is used to get rid of those mistakes. Anyway, then pattern formation morphogenesis, like getting rid of the webbing. Remember when we talked about insects, there are times when there's the cells that have only a transient function, like guidepost cells, where those guidepost cells I told you about, they're crucial for the sensory information in the insect leg to get its growth cone to the central nervous system, but after that they die because they've done their job. So you can take a look at some of these other possibilities. I'm not going to ask you to repeat any of them, but I do want you to know, well, you can satisfy your own curiosity if you want to see why some of the things that people have speculated about why they could be useful. Whether any of these or all of these are correct in terms of the function they serve, as I say, the one thing we know for sure, living organisms on this planet anyway, when they make lots of cells, they, even if they don't only make 1,000 cells like C. elegans, some of them they make only to destroy again. So one of the areas in which the concern or interest in apoptosis is really pretty strong is in amyotrophic lateral sclerosis, or ALS, also known as Lou Gehrig disease. And it's also sometimes called motor neuron disease. And the most famous person to have had ALS was Stephen Hawking, the physicist, who also pretty much holds the record for having survived the longest after getting a diagnosis, which was decades. And so he was diagnosed as a relatively young man, I think in his 20s, and yet he lived, I believe, into his 50s, 60s. Anyway, so he was the exception that proves the rule, because most people, it's a horrible disease to get, because what happens is you lose more and more control of your muscles, and yet mental functioning stays totally clear till the end. So imagine that you're slowly becoming paralyzed in your body, and you're totally aware of what's happening. So the statistic I always remember is most people, after they get a diagnosis, half of those patients will be dead in 5 years. Typically, the diagnosis isn't as a young person, but rather as middle-aged or older. And what happens is, when you do a post-mortem of a person who has died of ALS, the thing that's obvious is the muscles have all wasted away. There's this, they look skeletal because their muscles have lost both strength and mass over the years. And when you look at the spinal cord, almost all the motor neurons are gone. So they've died. As we've seen, for motor neurons to die on their own, that's a regular part of life. And so there's been a lot of interest that maybe the problem with ALS is that for some reason the developmental program of having motor neurons die has been re-triggered to disastrous effect. And that means there's been lots of interest in neurotrophic factors as a possible cure or treatment for this disease. There are 90% of ALS cases, we have no idea what happened, so-called idiopathic or cases. But there's a minority of cases where there are known genetic defects that are responsible for it. And one of these is an enzyme known as superoxide dismutase. And so people inserted the diseased allele for SOD into mice, and lo and behold, the mice started showing the sort of ALS-like symptoms of muscle wasting and progressive paralysis and eventually death. And so there was a lot of interest in this model. I'm not going to talk a lot about what superoxide dismutase does. In general, it's seen as something, as an enzyme that reduces the creation of so-called free radicals, which are known to be fairly destructive. And so you might think so in humans, when they have this superoxide dismutase allele, it's not taking care of enough free radicals, and so damage builds up and the cell dies. But that may not be the case at all, because what they found was when they inserted into mice, transgenic mice gave them the human allele in addition to their own superoxide dismutase. So they've got the allele too. Theirs is perfectly fine. If it's doing a job, it should continue to do so. But what they found is those mice got sick. So in other words, the superoxide dismutase was a problem of what geneticists call a gain of function. Somehow the human diseased allele has gained a function that the normal allele does not normally take care of. So it's not that it's failing to do something, but rather this allele is doing something, and it turns out to be harmful. What was interesting is, for many years, it was assumed that the problem in ALS was that the motor neurons die. In Britain, they spell it motor neuron disease. The idea was, since there's such a dramatic loss of motor neurons, the motor neurons die, and then the muscles waste because the motor neurons die away, and their axons retract, and the muscles, without innervation, lose their function and lose their mass. But there's growing evidence that, in fact, in ALS, the motor neurons are not the primary place where things go wrong, that it might be in muscles instead. And one of the things to sort of cause people to rethink this was they made some mosaic mice that consist of both wild type and mutant embryos, and they did it the old-fashioned way, just taking blastulas and physically agitating them together. In a minority of cases, they will blend. And then, so you could end up with a mouse that has some wild-type cells right next to some SOD mutant cells. And what they found is that the whole mouse showed disease, but when they looked in the spinal cord, wild-type motor neurons, those that did not have the diseased allele, were just as likely to die as those with the mutant allele. And so one of the things we know in mice, at least, that the gain of function that SOD causes to have the animals die is not acting in the motor neurons, because even if the motor neurons don't have the allele working, they still die. And so another possibility is that the glia is acting maybe in the spinal cord the SOD is normally acting in the spinal cord on glia, or a gaining possibility that things go wrong first in the muscle, and that that the muscles die first, and then the muscles, as they die, they stop providing neurotrophic factor to the motor neurons, and then the motor neurons die. So, I mean, you've all heard about ALS. By now, there have been several, important, fundraising efforts to try to fund more research into ALS. That you may have heard of the Bucket Challenge and things like that. And I gotta say, you really don't want any of your loved ones to get a diagnosis of ALS, because we've understood the nature of the disease for decades now, and the idea that there might be a neurotrophic factor that could be given that would either keep the muscles alive and therefore the motor neurons, or keep the motor neurons alive. It's been an enormous slag heap of disappointment. It's really quite perplexing how little progress there's been in understanding and coming up with some treatment for ALS. And anyway, it's a bad disease to get. So, and I'll tell you about one research program line that looked so exciting, and turned out to be for naught. So we mentioned briefly glial-derived neurotrophic factor. First of all, it turns out that the search for, if indeed muscles provide some neurotrophic factor that keeps motor neurons alive during development, no one's been able to quite find it. And so that was another surprise, that while NGF is so exquisitely important for sympathetic and DRG cells and other systems, other neurotrophic factors, we know that it's the one that got to have some of that to survive. No one's been able to find, a single neurotrophic factor that seems to be responsible for motor neuron death during development. And so people looked at things like BDNF or ciliary neurotrophic factor, things like that. And all three of these factors, if you look at how motor neurons survive in vivo, they all promoted a little bit in mice, but no one is really providing a sort of a convincing effect. It's like they all have sort of a marginal effects. And in mice, because in vitro, GDNF was even better than BDNF or CNTF at keeping motor neurons alive, that they decided to give GDNF at the muscle. And so they did experiments like this where they have the SOD mice, where eventually the motor neurons dies, the muscles waste, and the animal is doomed. And in some animals, they provided GDNF to the spinal cord and to see what happened. And in those cases, the muscles still wasted, the motor neurons still died back, and it was very ineffective. On the other hand, if they provided GDNF to the muscles instead of the spinal cord, then in the mice, that kept the muscles alive. And so there was, so it's the muscles that have to have the GDNF, and then they provide something, we don't know what, something else that is keeping the motor neurons alive. Or maybe it is that the GDNF is what's keeping the motor neurons alive, but it can only take it up through its axon terminals, not through its cell bodies when you put it in the spinal cord. So that then led to a lot of excitement about providing GDNF to people with ALS. Remember, these people are doomed, and so, of course, consent is very important, but they're, eager, these patients are eager to be involved in possibly finding a cure, or at least a treatment. And so they, in a study in people, they took people with ALS and they engineered cells that secreted GDNF, and they injected these people in both legs. They injected either cells that are secreting GDNF or control, so treated or untreated. And then the question was, look at how many motor neurons survive in the spinal cord after death, and they identify the motor neurons because they have choline acetyltransferase, the enzyme for dealing with acetylcholine, for chewing up acetylcholine at the neuromuscular junction. Anyway, and it's a fairly complicated graph here. In control patients, this is about how many motor neurons you'd see post-mortem, but in these ALS patients, and so each letter indicates a different patient, and the variable is compare the leg that was treated with the leg that was untreated, and what you can see is there's no significant difference. Here's one patient in which the treated leg in fact, had even fewer motor neurons than the untreated the untreated spinal cord. So that was, incredibly disappointing. And as I say, as of today, I think the FDA has approved 1 or perhaps 2 drugs that are hoped to maybe slow down the progression of ALS. But if so, they don't slow it down by much. And so if you want to get a Nobel Prize, by all means, figure out how, what's going wrong in ALS and come up with a treatment, and you'll bring, millions of people worldwide will have their lifespans expanded greatly. It'd be a great thing to do. I will say that one of the other things that experiments like this, where GDNF was so effective in the mouse model of SOD, but not in humans, is there's growing dissatisfaction or growing concern that the SOD mice, however cool they are and however much they replicate what looks like ALS to us, maybe they're not a good model of what goes wrong in people with ALS. And remember, these patients with ALS, the majority of them had a perfectly normal SOD allele, right? Because it's only 5 to 10% of ALS patients that have that mutation in SOD. And so it could be that, although still, if you could find a way to save the lives of those 5 or 10% of patients, that would be an incredible contribution. Now, so that's it for the chapter 7. And so this is what, we've covered so far. What's in the book are sections 7.1 through 7.8 inclusive. And what you should do now, when we have class, we'll have a Kahoot over this material because, everyone needs a chance for a lollipop. But what you should do now is, if you've read up to 7.8, including 7.8 in the textbook on perusal, now you should switch to the new assignment on perusal, what I've called chapter 7.5, hormonally guided neural development. And I'm going to talk about that next, and we'll just talk about that first portion, just what's in that document. And by the way, if things work right, perusal should let you make comments on this PDF I put in there, and so you have a chance to gain another 3 points towards your perusal, towards getting your perusal participation. Okay, so now I'm going to talk about what's in that new chapter portion. And when we're done with this, then you should go back to the perusal chapter in the book and read the rest of that chapter 7, which is, section 7.9, hormones, direct sexual differentiation of the vertebrate body and behavior, through the rest of the chapter. And of course, when you get back from your October break, we'll talk about that in class as well. So here, this is the new material that'll be in the second edition, about where there's a new chapter 8 that I'm calling hormonally guided neural development. So here are the learning objectives that are also, of course, in the notes that you'll see on D2L. And so in talking about hormones, just a little bit of terminology. So we call cells that release a hormone into general circulation that affects many different parts of the body, we call those endocrine cells. And often they're lumped together in an endocrine gland, like the testes or the thyroid or the adrenal glands. They release the hormone into general circulation. It goes everywhere. And there are cells scattered around the body that, if they have the right receptor to respond to the hormone, then we can call them a target cell. And of course, there are many cells that don't have the receptors respond to hormone and pay no attention. And And then we'll also talk about neuroendocrine cells. So a neuroendocrine cell is kind of a neuron and kind of an endocrine gland, hence the name. It's a neuron in the sense that it has ordinary synapses that make action potentials and the like. But a neuroendocrine gland, or sometimes called a neurosecretory cell, when the action potential gets to the end of the terminal and it releases its, exocytosis happens and chemical signals get released, they're not being released into a synapse. Rather, they're being released into general circulation and again can act on a target cell and have an effect. So we'll talk about several different kinds of hormones that are important in guiding neural development. The steroid hormones, like the estrogens and androgens, so estradiol is an estrogen. The steroid hormones all have in common this backbone that is taken from the diet, from cholesterol. Basically, it's the same 4 carbon rings structure that is found in cholesterol. And basically, you make steroid hormones primarily from, we animals do, from plant food where we get cholesterol, and then we do chemical modifications to add various different parts to this basic backbone to make different hormones. Estradiol being an example. We'll talk quite a bit about thyroxine, which is also known as tetraiodothyronine. It's one of the few products in the body that requires iodine, and that'll be important later. And you can see it's called tetraiodo because each molecule needs 4 of the iodine molecules. And we'll see that the structure of the thyroid hormones, there's 2. There's also a triiodothyronine, T3 and T4, it's sometimes called abbreviated. The structure is nothing like the steroid molecules, but in many ways the thyroid hormones act like steroid hormones, and as we'll see, there's reason to think that the thyroid hormones were there first. And we'll also talk about, in insects, we'll talk about juvenile hormone, which is, is a different chemical structure. I'm not going to bother telling you that, but it's very important in insects, not in mammals. And so for all the variety of hormones that are out there, there are basically 2 ways that hormones have influences on the body. Many hormones, typically protein hormones, most proteins cannot get through cell membranes. So most protein hormones, if they're going to affect the target cell, it's because the target cell has inserted into its membrane a receptor that recognizes a hormone on the outside, and then the binding of the hormone, that transient binding of the hormone, the ligand to the receptor, causes intracellular changes, often involving G proteins and the sort of things, and second messengers, the sort of things you've studied in other biology classes, and can have lots of effects inside the body, including, I'm not showing it here, but it can eventually cause changes in gene expression at the nucleus. Steroid hormones are a little different. First of all, they're tiny, so this is not to scale. Protein hormones are huge compared to steroid hormones. Also, steroid hormones are lipophilic. That means they dissolve readily in fat, so they can pass right through cell membranes. So when a steroid hormone is released into circulation, basically it goes through every cell. It gets to every cell in the body. But some cells are target cells because they've made a receptor, a steroid, that recognizes the steroid. And again, this isn't the scale, to scale. The steroid hormone molecule is tiny compared to the receptor. But once the hormone binds to the receptor, changes the conformation of this larger protein, and that gives it an affinity to bind to DNA. And in binding to DNA, it acts as a promoter or inhibitor or enhancer and can alter protein production or gene expression. In the regulation of hormonal release is itself almost a whole course, so we won't talk about it a lot. But I do want you to know about the differences and similarities between vertebrates and invertebrates. So in people, in mammals, in the vertebrates I should say, it is the brain that controls the release of every hormone. One way or the other, the brain is the organ that is gathering information, integrating information, and making a decision about when it is and is not appropriate to release hormones. So, for example, the vast majority of animals on Earth are seasonal breeders. They don't bother having their gonads be active all year round. It's the brain that's going to decide, is this the right time of year, the right place, are things safe enough, is there enough food, stuff like that, to decide whether to start making gametes. Anyway, and same for everything else in terms of food, water. It's always the brain that is gathering information and making a decision and controlling hormone secretion. Well, there are two basic mechanisms that the brain uses to control hormone release, both of which center around the pituitary gland. So the pituitary gland, which sits right underneath the brain and is attached to it by the so-called pituitary stalk, it has an anterior portion and a posterior portion, and the posterior portion is actually embryologically derived from neural tissue, whereas the anterior portion is not. And so they act in two quite distinctively different ways. For the posterior pituitary, we won't talk about it a lot, but it comes up some, and for completeness I want to talk about it. There are two portions of the hypothalamus, and by the way, I'll just say, the hypothalamus, which sits just above the pituitary stalk, it's crucial for controlling both the anterior and posterior portion of the pituitary. For the posterior pituitary, there are two nuclei in the brain, the supranucleus and the paraventricular nucleus, that are chock full of neurosecretory or neuroendocrine cells, and they send their axons down through the pituitary stalk, and then they reach capillaries in the posterior pituitary, and they release their product directly there, and those products serve as hormones. And the two most famous ones are oxytocin and vasopressin. Oxytocin is important for childbirth and also playing a role in lactation, and vasopressin is crucial for controlling fluid intake and the amount of fluid in your body. But both are also very much involved in social behaviors, and so there's a lot of interest in these two hormones in terms of how it is they the role they play in acting on the nervous system, not just the body, but also in the brain to influence social behaviors. But we won't be talking about that very much. What we will be talking about is the anterior portion of the pituitary, where things are quite different in terms of the control. So again, the hypothalamus is controlling the anterior pituitary totally, but the difference is that for the anterior pituitary, there are scattered neuroendocrine cells throughout the hypothalamus, and we're just showing a few here as a cartoon. And these cells make synapses, make action potentials, and all that stuff. And when they release their product, they don't release it onto a synapse, but rather release it into this arterial source here, in the so-called hypophyseal artery. Hypophysis is just another name for pituitary. I won't use it much. And so they release their products here, and the product they're releasing is a hormone. And the hormone gets into this special capillary bed that goes from the median eminence of the hypothalamus, goes to the anterior pituitary, and now their hormone acts on hormone-secreting cells, endocrine cells in the anterior pituitary that release a hormone in their own turn. So the terminology is the neuroendocrine cells in the hypothalamus, they produce releasing hormones. And the releasing hormones travels down the hypothalamic pituitary portal system. The system is called a portal system, as in a porter, someone who carries things for you, because it's going to carry the signal from the median eminence to the anterior pituitary. And then the pituitary cells in turn, and by the way, these releasing hormones, if we look in the blood and try to look for them, they are so rare that you can barely detect them. Usually you can't detect them because their action is quite local. by the time they've gone through the anterior pituitary and gone into general circulation, they're so scarce as to be almost undetectable. But the anterior pituitary cells, they release other hormones, which are known as tropic hormones. Tropic because they are heading for different glands. Now, of course, in fact, they go everywhere, but there are only certain glands that are going to be responsive to them because they're the target cells. So that's one way the brain controls those hormones. It controls directly the release of releasing hormones, which controls what the anterior pituitary releases. And here are some of the tropic hormones that you might have heard about that we're going to talk, some of which we're going to talk about that are involved. Prolactin to promote lactation, gonadotropic hormones, FSH, follicle-stimulating hormone, and luteinizing hormone that control gonadal function, thyroid-stimulating hormone, adrenocorticotropic hormone, the hormone from the anterior pituitary that stimulates the adrenal glands to make adrenal steroids, and growth hormone, which acts throughout the body and promotes development everywhere, hence the name. By the way, once people discovered the pituitary and discovered that if you destroyed the pituitary or cut this link between the pituitary and the hypothalamus, they noticed that all the different endocrine glands around the body all shrank away. And the animals, it was difficult to keep the animals alive when that happened. And so in the mid-20th century, people would talk about the pituitary as being the master gland because when it was destroyed, every gland in the body seemed to get tiny and withered. Well, and it is a master gland in that way, but believe me, this master gland is itself being totally tightly controlled by the hypothalamus lying above it. So that's how those are the two main systems of hormone regulation in mammals and vertebrates that we'll talk about. In insects, there's some similarities and differences. So insects also have neuroendocrine glands. In fact, they were first discovered and realized how they worked in insects. You'll not be surprised to learn. And things that are kind of similar to the mammalian system. So some neuroendocrine cell brains send their axons to what's known as a portion connected to the brain called the corpora cardiaca. Doesn't matter why it's called that. And they're acting like posterior pituitary cells because they release their hormone directly into circulation there. And so this particular neuroendocrine cell is releasing a peptide hormone called PTTH. I don't want you to. You can guess why it's called that, because it acts on what's known as a prothoracic gland. And the prothoracic gland, in its turn, in response to this hormone that came into general circulation, like oxytocin or vasopressin, it produces a steroid hormone in response. A steroid hormone known as ecdysone, which is not seen in mammals, but it acts like other steroid hormones in mammals. And it's called ecdysone because it's important for telling the body to make a new exoskeleton. Remember, insects don't grow internally. Insects have a rigid outer skin, the exoskeleton. And in order for them to grow, they have to grow a new skeleton that's soft underneath the old one, and then eventually break the old one and come out. And so that business of coming out of a new skin, that's literally called ecdysis, shedding an old skin. And so that's why they call it the hormone ecdysone. So on the other hand, there are other neuroendocrine cells that provide a releasing hormone that act on a different body, the corpora allata. I don't need you to remember the names corpora allata, corpora cardiac. I want you to recognize them. But anyway, and there it acts on endocrine cells in the corpora allata that release a different hormone in turn. And in this case, we're focusing on one called juvenile hormone. And it's called juvenile hormone because as long as juvenile hormone is in circulation, the animal will not, when it undergoes ecdysis, take on an adult form. So if in the first larva comes out, when it starts to make a new body, if an ecdysone's going to come eventually and tell it, okay, break out that old skin and come on out in your soft skin, which will soon grow hard, but it's a bigger skin. And as long as juvenile hormones are around, each time the body will undergo taking on a new larval instar. But once juvenile hormone starts to decline, now when the ecdysone comes, the body interprets the signal very differently. So in insects like moths and butterflies, where there's complete metamorphosis, what happens now is indeed the last larval instar makes a new skin inside, but it's not a new larval skin, it's a pupal skin. And so when the signal from ecdysone comes and says, all right, break the old skin and come out, it emerges as a pupa with not much in the way of being able to move around. And in the meantime, the larva has gone somewhere. Maybe it's spun a cocoon or chrysalis, or maybe it's gone underground. And now the pupa sits protected for a while. And now, again, with no juvenile hormone around, it begins making the adult body inside the pupal skin. And when the next round of ecdysone comes, it comes out into its adult form. And by the way, there's a special word for this particular ecdysis from pupa to an adult. It's known as eclosion. So it has, come out of the closet, if you will. So it's eclosion. Okay? Now, there's interesting things that happen. Well, first of all, I'll just point out that these two hormones, ecdysone and juvenile hormone, they offer an excellent example of what hormones are great at, which is sending a signal throughout the body and having different parts of the body take very different paths in response to that hormone. It's a great way of coordinating a big change. How big a change? Like the change of having a larval body. Maybe, by the way, it's an aquatic creature in case of mosquitoes and things like that. So that tell the different parts of the body, don't make what you need for a new larva, make what you need for a pupa, and then to tell the pupa, don't make another pupa, make an adult. Well, you can look at the differences. I mean, look at all the things that the adult has that the larva and the pupa don't. So that requires a lot of coordination. Coordinating that different parts of the body take on totally different fates than what they had before. And, hormones, because they go everywhere, they're ideal for sending that sort of a signal. So here's another look at it, right? So now we're looking at the Manduca sexta. It's the tobacco hornworm or the tomato hornworm. If you've grown tomatoes, you've seen the larvae there. And if you're like my family, you're taught to pick every larva off and step on it to get rid of them, because they will eat up your tomato leaves. Anyway, when juvenile hormone is high, it goes from one larval instar to another. And in Manduca, typically, here's the peak of ecdysone that's telling it to come out with a new body. But so it makes the fifth larval instar. But then juvenile hormone levels decline and stay low the rest of its life because it's no longer a juvenile. And then there'll be one little release of ecdysone that will commit it to making a pupal body. And then when the next big rise in ecdysone comes in the absence of juvenile hormone, it undergoes ecdysis as a pupa. So it's been making a pupal skin all along while it's continuing to eat your tomato leaves inside the larval skin. And then the pupa will have a very complicated release of ectosteroids that, in the absence of juvenile hormone, is telling all the different parts of the body, time to make an adult. And then eventually what will happen is it will eclose and into an adult. And here, by the way, is a look at the ecdysone. You can see that same basic 5-carbon ring backbone that's in all the steroids. Well, I'm talking about Manduca because there's something interesting. As you know, all insects have 6 legs, but if you look at a Manduca, it looks like it has, 6 legs up here and 8 legs back there. But these aren't real legs, they're prolegs. So, and they're very simple. They have one muscle fiber that connects to the end of the proleg and connects inside the body. And so they can only retract the leg. And why does the leg go out? Well, it's just the body pressure inside the animal pushes them out, right? And each proleg muscle fiber has a single motoron that innervates it that's found in the nearby abdominal ganglion. And the motoron looks like this. And so here's the cell body, here's the axon that's going to the muscle, and then here's a very complex dendritic tree of this motoron. And of course, they're simple legs, but the animal has to control them in order to locomote. So it uses these prolegs to help hold on to the stem, and for it to go forward, of course, there's got to be coordination of moving the true legs at the front and having the prolegs in the back let go. So this is the muscle happens to be called the plantar retractor muscle, okay? Well, so it's only the larvae that have prolegs. Pupa and adults don't need them. And so interesting things happen. First of all, there are, 4 different prolegs, A3, 4, 5, and 6, and each have their motorons. And so, they all look pretty similar. They're motorons. And when that peak of ecdysis comes in the 5th larval instar, when juvenile hormone levels are low, when that happens, the dendritic trees remodel, because, of course, they're going to behave differently in the pupa than they were before. And some, when the adult body is getting made, some of these motorons in A3 and A4, they survive and take on a totally different function. Now they're not affecting the retractor muscle because it died. It underwent apoptosis. Now these motorons are involved in circulation. And some of the motorons in the 5th and 6th abdominal segments, they die. And at eclosion, these motorons that survived into the pupa to play a circulatory role, they've now lost their function and they die. And all of these decisions about whether to die here or whether to die there, they're all being controlled by the interaction of ecdysone and juvenile hormone. So for example, one way to show that is you might ask, well, are the muscles dying and therefore the motoron dies? Or is it the motoron that's dying and therefore the muscle dies? And the answer is quite clearly that they're listening independently. Each of them is independently listening to juvenile hormone and ecdysone and making a decision whether to die now or to die later. So one way you can do that is, in one experiment, they applied juvenile hormone just to the prolegs. And so here's a pupa that developed, and it's got a pupal skin everywhere except at the skin there where juvenile hormone is present. And they asked the question, well, what's happening in these animals? What happens to the muscles and motorons? But it turned out that the muscle died anyway and the motoron died anyway. So again, it's like every cell in the body is listening to these two signals, juvenile hormone and ecdysone, and making their decision about what sort of changes to make based on those two signals without, and they don't seem to be paying any attention to what the rest of the body is up to. And so this is, another indication of how great hormones are at coordinating things. And that's their great strength, is they can serve as a coordinating signal throughout the body to have the body undergo changes that are coordinated, different changes in different cells, but all towards the same end. In the case of insects, towards, growing and becoming an adult and reproducing. And in the case of vertebrates, telling the body, make changes during the seasons, changes during the time of day, changes across the year, changes in terms of growth about, for example, is it time to reproduce? All right, well, so that's enough about that. We'll take questions about that when we meet after the October break, and then we'll start talking about another hormone, go back to vertebrates, and talk about thyroid hormone. So see you then. Hope you're having a great break.